Thank you very much. So today you treat this as light entertainment. This is nothing sort of in-depth scientific, really, just light entertainment. And I'm going to talk to you about time traveling or using certain aspects of biology as vehicles to travel back into time, to ask questions about origins of nervous system, and also to ask questions about whether what we look at today is ancient and has been around for a very long time, which will help us to understand it, I think, or whether they are recent innovations, and we're looking at evolutionary innovation within the last, say, 20 or to 50 million years. People looking at Drosophila are looking at a very strange organism, really, because it hasn't been around very long, about 50 million years, which is an instant, really, in evolution geological time. So are we studying the visual system and nervous system of Drosophila as a latecomer, or does it really represent something that evolved very early in metazoan life? And if it evolved very early, it's more likely to share attributes with other branches of the evolutionary tree, which also are deep-rooted. Again, Dobzhansky's lovely caution to us all that nothing in biology makes sense except in the light of evolution. I think probably nothing in science really makes sense either. Compound eyes have been around for quite a while. In organisms, they started off in organisms like this, which is about 535 million years old. And through geological time, we have end up with, from this sort of stem group taxa, we end up with these magnificent arthropods, both marine and also terrestrial, and some of them which can fly and which can catch, I'm sorry, Srini, which actually hunt bees.
Not good.
Not good at all. These are really extraordinary creatures. They have these ballistic flights. They're extremely accurate in their predation. So what has changed from here to here? And that's a question, because presumably not only this animal can see, of course, objects and see objects against the background and must compute then visual motion, but it is likely that this creature too was able to see some motion at least to avoid predators, if it wasn't a predator itself. Now, when we look across the arthropods, the insects and the crustaceans, and insects have evolved from the crustacean ancestor, we can ask the question, what was the simple system, what is the more complicated system? And amongst the crustaceans, we have two major groups. One is called the Brachiopoda, and the other called the Malacostraca, and plus the Insecta, we call them the Pancrustacea. And these two groups differ in a very, very interesting fashion. The brachiopods have retintopic neuropils, and they have two of them only. They have two of them. And there's no crossing over of the projections of the first neuropil to the second neuropil. So this is the laminar, and this is the lobular plate-like neuropil, a tectum-like neuropil, which is equipped in many brachiopods with wide-field neurons, rather like the wide-field neurons in the fly lobular plate that we discussed 2 days ago. But insects and malacostracans are very different indeed. They have three nested neuropils, 1, 2, and 3, and sometimes a fourth, which is budded off from the third. And these neuropils are connected by chiasmata, crossing axons, so that the linear organization of neurons at this level is simply reversed at this level and then re-reversed at this level. And this means the polarity of the neurons, too, are very different between brachiopods and malacostracans. And for many, many years, well, not for many years, indeed until, into the present, there are two schools of thought. One school insists that the brachiopods, because they're simple, gave rise to this more complex condition, and that brachiopods gave rise both to the malacostracan crustaceans, and brachiopods are the ancestral group of the insect. So the insect ancestor was a branchiopod-like organism with such a simple nervous system. You can ask any kind of questions you like. Yes.
What is the reason for? Is there any functional significance to this reversal and re-reversal?
There's no functional significance. It's actually an accident of development. The medulla and the lamina develop from two sets of stem cells. And as they develop, axons have to cross from one side to the other as they develop. And once you have one crossing axon, the other, it acts as a pioneer fiber for the next outgrowth of the next receptor, photoreceptor, receptor number 8 actually, which then crosses over the pioneer fiber and crosses over the pioneer fiber in sequence.
It requires you to have longer axons and everything, so it doesn't seem like a good design.
Not really, but when you look at the axons, the ones that actually have the shortest length are the thinnest, and the ones that have the longest length are the fattest. So they do try and balance the speed of conduction, I think, across the whole array. Yeah. So you can actually go from here to here. If you double up this neuropil, you double up the stem cells, and you split it into two neuropils. There's a way of doing this. But for the sake of our considerations, 2 optic neuropils, no chiasma, inverted second order relays, and then here 3 optic neuropils, 2 chiasmata, and then centripetal relays. So which is the ancient condition, which is the modern condition, and does this tell us anything about circuits that we study today? So, as I said, this is entertainment. We're not going to go into real, hard neuroscience here. I'm just going to try to illustrate how we can show that this can't possibly be the precursor condition of the malacostracan insecta. So visual neuropils, there we are, entomostracans or brachiopods 2, malacostracans 3, or sometimes 4. And the real problem here is that for these new neuropils, if you like, new neuropils being those that don't exist in the brachiopods, these new neuropils are the medulla and the lobula, which are composed of a huge number of cell types and varieties. And as we've seen 2 days ago, this structure, this medulla here, is really important in that it provides the outputs and probably the computational circuits for elementary motion detection that occurs across neighboring armatidia. So does this mean that the circuits for motion detection in malacostracans and insects are recent evolutionary innovations? It's difficult to think that they might be, but could they be? Or could there be so many circuits for motion detection that what we're looking at today as important with regard to visual flow fields is actually a recent innovation that's associated with the control of flight? We have no idea. So it's quite important to get to the basal ground plan, the evolutionary ancient ground plan of these systems. So we have 2 scenarios for the evolution of the insects and the malacostracan crustaceans, the kind of crustaceans that you and I like to eat in our paella. There's these guys, so these are the malacostracans, and one can envisage an ancestor of a malacostracan-like ancestor that gave rise to the malacostraca and to these brachiopods, and the brachiopods have suffered an evolved reduction and loss of characters. And then the insects here. Or there's this ancestry of a simple branchiopod-like organism which has given rise to the brachiopods and the malacostracans, and then the other guys here, the insects. And it suggests then the insects must have arisen from the malacostracans in both instances. So there's a problem. How do we go about resolving this problem? And can we do it just looking at extant species? Well, you've seen this yesterday, the strategy of using neurocladistics. So I just remind you again, we can use neurocladistics to interrogate the cladograms regarding what features are common or not common across a multitude of taxa. And what we're looking at here is the organization of the visual systems. So here is a primitive insect, an Archaeognathan. It has its 1, 2, 3, and 4 actually visual neuropils. Here is a cockroach, 1, 2, 3, 4 visual neuropils. This is a rather nice, splendid specimen of a lobster. It has also the 4 nested visual neuropils. This is a very interesting animal. It's a copepod. They're tiny little creatures. which live everywhere. They're extraordinarily common animals. You can even find them in glacial water in the Himalayas. At high altitudes, the glaciers have a very thin sheet of water in the summertime underneath the ice, and you find these animals swimming around, eating presumably bacteria or goodness knows what. They're the largest biomass on this planet, and they're at the bottom of the food chain for almost everything that's in the sea. They're very interesting because the midbrain of these animals is exactly like that of a lobster. So these basically are malacostracan crustaceans that have lost vision. They live in an extremely simple environment, ecology, and they have no need for vision. So they've lost the vision. But you can actually determine that some of these blind animals are malacostracans. And then there's this thing here, which is a branchiopod brain with its 2 optic neuropils and a very, very simple organization indeed. The central body complex, which we discussed yesterday, is very reduced. The olfactory lobes are almost non-existent. There is no mushroom body or hemiellipsoid body in any branchiopod. And more importantly, the third ganglion of the brain is situated well behind the gut, whereas everywhere else, the third ganglion is partially fused with the second ganglion of the brain, and the gut penetrates the third ganglion. So this is a very important morphological distinction between the Brachiopoda and all other insects and crustaceans. I've included here a phylogenetic tree which is derived from express sequence tag analysis by my student David Andrew, who is in my laboratory. And this tree overlaps beautifully it's another one of the molecular trees that overlaps beautifully with the morphological tree gained from these neural character reconstructions. And in both cases, the brachiopods appear here, slap bang in the middle of the malacostracan insect ensemble, which suggests that brachiopods are not, they can't be ancestors, or they cannot be a branchiopod-like ancestor to the malacostracan crustaceans and the insects. So this brain presumably has evolved due to loss and reversal of characters. And this aspect of loss and reversal is one that's very much ignored by many evolutionary biologists. And if you only use molecular cues, molecular traits, you can never actually detect reversal and loss in the phylogeny. You have to combine morphology with molecular phylogeny in order to understand this kind of phenomenon. Okay, so here then is the branchiopod type. Yes?
On the previous slide, so you're saying that the data that we're looking at here would be showing us this a reversal.
Yes.
Right? So how do I really see that here?
You see the reversal. You see a loss. You see a loss. One must assume that was a simple precursor, very in deep time. One must assume that. Okay. But we cannot say that from this organization, we cannot claim that the branchiopod-like structure would be the ancestor. However, we must assume there's a simplicity that preceded the complexity. So what we have here is
important heuristic here. You always
yes. Well, unless the complexity appeared suddenly, like some kind of Goldsmith, hopeful monster in deep time. But maybe it did. We'll see. Okay. So anyway, this simplicity suggests a reversal, but certainly even if it's no reversal, there's clearly a loss of structures here. Okay. So and if we go now then to the actual neuropil, here then is an example of a branchiopod optic lobe. There's the lamina, and this is the lobular plate-like structure, which we call the optic tectum, at least just for the sake of distinguishing it from the lobular plate of a crustacean or an insect. This is the insect organization: lamina, medulla, lobular plate. Here is the crustacean: lamina, medulla, lobular, and this is the lobular plate-like structure here. And the lobular plate always has uncrossed axons between it and the medulla. This is just a sort of a taster, if you like, showing that the medulla it contains these beautiful transmedullary neurons with very characteristic shapes. And so, there's no prize really available, but those people in the audience that might suggest which of these comes from a crustacean, a malacostracan crustacean and an insect, are doing really, very well. You really can't tell the difference. So the organization and the shapes of these neurons in insects and malacostracan crustaceans, it's almost identical. It's highly conserved.
At what level would you start to see differences?
Receptor that's a good question. No, you see the differences down here at the level of the lobule and the lobular plate.
Okay.
This is where evolution is exerting itself, or expressing itself. So the way that information that's computed at this level is collated and collected at this level is very, very different across different taxa. Okay. That's actually rather interesting. So now you have then the elaboration of a malacostracan insect optic lobe can be from, if you like, from a branchiopod-like organization, sorry about the misspelling, or an evolved reversal and loss of optic centers in brachiopods from a more elaborate malacostracan-like ancestor. They're both equally plausible in terms of the evolutionary scenario. So here is the one where we have the intermediate and the branchiopod as a derivative of what probably was a malacostracan-like ancestor, and here we have the branchiopod-like ancestor to everything that is insect or malacostracan. Okay. But there's another group of arthropods which are really, really fascinating. And these are the centipedes. And these centipedes are more like the fossil I showed you in the first slide. They have very nicely differentiated head. And all the other segments pretty much are homonymous. They are just an iteration all the way down the length of the body. And the centipedes, most of them have rather miserable visual systems, but the most primitive group of centipedes, called the scolopendromorph, the scutigeromorphs, these things here, which are fantastic animals. They are highly efficient predators and very beautiful. In the tropics, they get to about this big, pretty fearsome creatures. They have a lamina and they have a lobular plate-like structure, which is equipped with beautiful giant tangential neurons, the axons of which extend to the midbrain, where they terminate on premotor descending neurons. And you put this animal into a striped drum, and it's extremely frustrating. It'll move with the stripes, and then it'll get bored and move opposite the stripes. So you have a sort of plus minus zero result. But they are very, very excited by moving stripes. And so we think these are motion detecting circuits here. And if they are motion detecting circuits, then what's with these T4s and T5 neurons we've been discussing 2 days ago that we believe are the elementary motion detector relays? Because there is no medulla here that could accommodate such small neurons. So the ancestral condition might be represented by today's scutigeromorphy. These are primitive mandibulates which are obviously branched off the main stem lineage that gave rise to the crustaceans and insects. And they have, like a branchiopod, they have a lamina and this tectile-like structure, which is connected by uncrossed axons. So maybe motion was the very first visual modality that was encoded by the earliest visual systems. So would we expect to find this, in the depth of time? So again, we can now suggest that the scutigeromorphs, or scutigeromorph-like ancestors, gave rise to the common ancestor, to the branchiopods, the malacostracans, and the insects. So this then would be the if you like, the Ur visual system. But how ancient is ancient? How far back do we have to go to find this? And the search image is then obviously clear. This is the search image. This is the Ur system, a lamina and uncrossed axons to a lobular plate-like structure. That has given rise to this more complex system, where what remains of the original system is the lamina and this visual tectum or lobular plate. And what is new is all this garbage in here in the outer and inner medulla. And it's thousands of neurons and hundreds of different kinds of neurons, and then the lobula, which is a very, very novel system altogether. We assume that it's novel. And we can also envisage that from the simple system, you can elaborate these columnar arrangements by adding, if you like, adding stem cells and adding repetitive division of lineages, of neural lineages to achieve greater complexity. It's not difficult to imagine, and in fact, there's a lot of evidence this is occurring in the central body complex. And in some insects, you have 4 or 5 cycles of division of neurons that extend into the columns of the central body complex. And in other insects, you have maybe 30 or 40 cycles of division, which provide many, many, many more cells in addition to the ground pattern. And Heinrich Reichert in Basel has been looking at this in great detail. In the mushroom bodies too, the number of divisions of the stem cells giving rise to these parallel fibers, the globule cells, is very different in different insects. In Drosophila, you have only about 1,200 neurons in a mushroom body, but in the honeybee, you have 350,000, and that's because the stem cells continue to divide and divide and divide, and to make many, many more ganglion mother cells and therefore neurons. So this is a very simple way of actually increasing complexity of the visual system, and one can imagine, one can speculate that it was driven by the increase over evolutionary time of the visual ecologies, both with respect to the structures and also with respect to the actual interactions of organisms of predation and predators. But that's pure speculation. But what we do know is when we go to modern crustaceans, where we find pelagic malacostricans living in the water column, which has very, very few visual cues, we see a remarkable reduction of the visual neuropils. They're incredibly reduced. And in some animals, which are ankyline, means they live in lightless grottos, such as this creature here, there are no visual systems left at all. There's no retina, there's no visual neuropil, and the same is true with the copepods. So we do know if you remove the visual ecology, then basically you are removing the necessity for a visual system. And since brain is extremely expensive, why keep it? Expensive metabolically, at least.
But Nick, if you would not just talk about, let's say, complete darkness and some form of sunlight, but you go to a measure of, let's say, the complexity of a visual ecology, how would it then scale?
Yes, this is something that really hasn't been looked at in great depth. Some of these insects that catch bees, these ballistic missiles, they seem to have very, very few cues that really interest them. There are sticks and there are objects moving across the visual field. They have actually a greatly reduced lobular plate. They've thrown away neurons that respond to vertical motion, partly because the bodies are very long. They have extremely good aerodynamic properties which obviate pitch control. And also the medulla seems to be much simpler than say, in a Drosophila. So this is an area where one needs to do some exploration, I think.
But what would be your prediction?
My prediction?
On how these structures would in a more subtle way start to vary if, let's say, we start to remove certain subsets of this visual ecology to cues.
Yes, I think the thing to do is to go into some of the aquatic insects because there are very, very nice, very discrete kinds of ecologies in the water, and there's a whole host of aquatic beetles that occupy these various ecologies, and then do a cross-taxonomic comparison. So maybe I will persuade some poor PhD student to study this, and of course will ruin his or her future chances of a job in America. You have to come to Europe. Right, okay.
But are you saying that then you would predict that most of the variations would occur in, let's say, the lobular plate?
I would predict most of the variations would occur in the lobular and the lobular plate. That's where we're seeing differences.
Right.
That's where we're seeing differences. So how old is the compound eye, and how elaborate was the earliest visual system? So the neural and molecular statistics suggest that the ancestral visual system comprised at least 3 nested centers. The ancestral visual system of the insects and the malacostran crustaceans. So the conclusive evidence will be provided by an ancient fossil of an ancient visual system, right? So can brains fossilize? And it's interesting that there is no compelling evidence yet, at least that I know of a fossilized brain from any vertebrate material. There are some suggestions of fossilized brains in some fossil fish, but those are actually then what remains when the brain is gone is the empty cranium, if you like, of the fish. So I started pursuing a line of research that I called paleo neuroscience. And the premises are this. So we have then two major evolutionary trajectories of the arthropods. One which gives rise to these things called the paradoxopoda. Those are the chelicerates, the limulus, the scorpions, spiders, and their ilk. And there's the others called the tetraconata, which are the Malacostra insecta and the mandibulate arthropods. And these are the crown group taxa which we can see today. And these are all the stem group taxa that existed once upon a time in deep time. And the stem group taxa are extinct, but they probably have features of these animals, or combinations of features of these animals, and features which no longer exist. So we have pretty good search image for the kinds of morphologies we would like to find. So one of them was this. So in 1908, Walcott went up to the Western Canadian Rockies, and he found a huge amount of fossil material which was ascribed to the Cambrian era about 500 million years ago. And a lot of these fossils have been described in some detail, but nobody actually has asked the question, how complex might their nervous systems have been? And the reason nobody asked that question is that nobody thought you could possibly see a nervous system in such a fossil. On the other hand, fossils are equipped with lots of skeletal pieces, otherwise you wouldn't see them, and these pieces include receptor organs, sensilla, facets, what are clearly olfactory receptor organs. So by looking at the sensilla and the organization of sensilla and the organization of the limbs, one could possibly gain a fair idea about the complexity of the ganglia that received the sensory inputs and that provided the actuators of the limbs, the motor circuits. And that's what I intended to do. So I went up many times, to the Smithsonian and grubbed around in their stacks. Somebody looking very carefully over my shoulder, didn't take any, and looked for a taxon which would seem to be a good specimen for investigation. And there's this one taxon called Waptia fielddensis, named after Walcott. No, after Field, after the village in which he found all these specimens, the village in the Rockies. And nobody described this in any detail. And it turned out that there were several groups, all thought each other were describing this fossil. And they were all very wary of treading on each other's toes. But I was a neuroscientist and had no idea of the social tensions that exist amongst fossil people. So I grabbed hold of this and reconstructed it. And it's a really very fascinating animal indeed, because it has attributes of not only crustaceans, but also insects. It has one pair of antennae, which are characteristic of insects, and actually of centipedes and millipedes, whereas all crustaceans have a first and second antenna. So the second antenna has gone in this creature. It also has a very nice pair of eyes. And the antennae are fascinating because the first two segments of the antennae seem to be naked of astrotasks. When you look at the rest of the antennae of this animal, you have then these beautiful collateral spines with rows of very, very short sensillae, which the organization of which is characteristic of a modern malacostracan crustacean, where you have rows of sensillae called astrotasks, which are the chemoreceptive hairs on the first antennae. In brachiopods, you have the first antennae, which are miserably equipped with just one or two astrotasks, completely different from a basal malacostracan and completely different from Waptia. So that was the first surprise. The antennae of Waptia looked like those of Nebalia, a basal malacostracan crustacean. So one might suppose, oh yeah, here we are. Here's the actual, the goods. Here's the Nebalia, the malacostracan antenna, showing this long mechanosensory hair, and then there's rows of very short, much shorter bristles, the aesthetats, the chemosensory hairs. And this is Waptia, the long flanking bristles, and then this row of shorter hairs. These are marvelous fossils, actually. They look like low-pass scanning electron micrographs if you put the fossil in water, and then which they didn't like doing actually in the Smithsonian, but it doesn't harm them. And then you use high-resolution binocular microscope, and it's really quite wonderful. You can see the mouth parts and everything. So one might assume that because Waptia and Nebalia are so similar in respect to their frontal morphology, that Waptia would have a well-developed olfactory system, as does Nebalia. This is the olfactory lobus of Nebalia. They're really gorgeous. And Nebalia has also rather a simple set of rather simple retina with rather few ommatidia, but it has a very clear lamina, medulla, and there's a lobula just down here. But one has to do more than that. One can't just simply extrapolate into a modern taxon. So one has to then go back to the fossil and ask, well, how much can one resolve on this creature? For example, here is one of the piece of the antenna, and this is a mechanoreceptor on the antenna, and you can actually see the point of articulation of the receptor. It's really quite remarkable. So these fossils are highly revealing, and best of all, some of them show what is clearly something in here, an outline of a structure. Now most paleontologists, when they see something within the fossil, a sort of outline of some organ, they say, oh, gut. Gut. It's got to be gut. Because they don't understand that gut is a much less dense tissue than brain, and brain is more likely to be preserved if it's flooded with minerals during fossilization than maybe the gut. And here we have clear evidence of an optic lobe, an optic stalk, a midbrain, and at least one side of the connectives leading to the thoracic ganglia. So then when we reconstruct that, here is the brain of Waptia. Possibly two optic neuropils, certainly a very nice antennal nerve, and then a fusion of material in the center here, and no evidence really of the next ganglion, unfortunately. So that is as far as one can get with Waptia. It's not terribly satisfying because it doesn't really tell us how many optic neuropils there are in the brain. It doesn't really tell us how many segments comprise that brain. But there are other sites. Yes?
Explain how you get from that middle image to the bottom one.
I'm just taking this and making a mirror image. That's all. The first arthropods were homonymous. This is something called it's called a lobopodian. There are lobopods alive today in Gondwanaland, bits of Gondwanaland which have become southern India, South America, Australia, New Zealand. They're called velvet worms, which are probably actually highly derived. But the earliest ones were certainly homonymous. They had a head, and that every single segment was identical afterwards. Here is the one I showed you in the beginning. There's a head, a pair of eyes, everything else is pretty much identical except at the end here. But this organization has persisted even until the most recent insects and crustaceans, because when you look at a developing Drosophila, you see that the developing ganglia, the thoracic ganglia, are homonymous. They're an iterated organization. It's only later on, when this thing undergoes metamorphosis, that some of the ganglia, the neurons of some of the ganglia proliferate, or the stem cells proliferate much more than others, and there's programmed cell death trimming out the neurons from other ganglia that don't serve, for example, an appendage. So this system evolves, if you like, to this organization using rather simple criteria. But the place to find the best fossils is here. This is Mao Hat Mountain. Mao's Hat Mountain, Mao Tian Shan. And it's in Yunnan province in China, and they're all centered around near the town of Changjiang, and these fauna provide the very best preserved earliest metazoans, including the two I've shown you here. And there's a piece of another one. And this is a really a lovely part of China, actually. Yunnan is gorgeous, a gorgeous part of China. And this is one example of one of these fossils, which I didn't actually find in Yunnan. I have to confess, I actually bought this fossil at the Gem and Mineral Show in Tucson. The fossils are sometimes enter the black market, if you like. And I thought it was cheaper buying it than flying to Yunnan. That was about 4 years ago. I've changed my mind since. Anyway, this is a very beautiful creature. It's about 4.5, 5 millimeters long. A great amount of detail of its organization. The sensory bristles on these pleopods that provided it with locomotion. It has sort of an eye stalk with lenses here, and then various other limbs. So it's a very nice specimen, but not really the best for looking for brain, because you really need a specimen which provides a top-down view and an inside. So a specimen like this. This is something called Fuxianhuia pratensa. It is not this big. It's about this size. A splendid animal, and it's rather a common. You get rather a lot of them. This is my wife, Camilla, on one of these quarries, and underneath her feet there must be several thousand fossils. You know, it's very tempting to go back there and blow the place up with dynamite, which people do actually, and then look through the shards. So this is a 635 million year old mudstone shale, which is very rich in fossilized material. So this is a super animal with a head shield, a rather simple organization. It's a very simple looking animal. It looks as simple as some of the brachiopods that exist today, with a head capsule and then eye stalks. And there are others too called Naroe, and I'll show you those in a moment. Now, when one looks at these fossils, one looks for a flattened specimen that has actually been split. That the actual fracture plane has gone through the body, and gives rise then to two parts called the part and the counterpart. And if the counterpart contains the gut, the preserved gut, then it's going to obscure the ganglia beneath it. And if it's upside down, if this is the dorsal, then it would obscure the brain. But you do get occasional fossils where the counterpart has actually been fractured such that the gut, or anything solid and large, has been lifted off the underlying structures, such as ganglia, or other way around, you know, if you invert it, then such as brain. Here is then an example of a specimen where the part, when you look down on it, actually is filled with gut. There's a nice antenna here, and you think, oh my goodness, this must be brain and this must be the thoracic ganglia. It turns out that this actually is not. These are diverticuli of a gut that extends all the way down the body to the anus. So that's disappointing. On the other hand, occasionally you get a fossil where the gut has been removed, at least in part, and then you can then find rather interesting structures which are obviously ganglia. And these are preserved ganglia. They're fossilized ganglia. And that is very, very exciting indeed, to see fossilized nervous system. But as I showed you about 5 slides ago, these reiterated ganglia are to be expected if you have a reiterated body structures. What one's looking for are specialized ganglia which actually form brain.
Nick, you said 635 million years.
635, yes.
That's into the Precambrian.
That's lower Cam. So 500. Did I say 600? That was a misspeak. 535 million years. I think it was actually written. Was it written 600?
It was.
Yeah.
It was.
Yeah.
Very stupid of me. It's 535 million, which is lower mid-Cambrian. But then Fuxianhui provided some really interesting and exciting data. This is one view of the head. And here are sort of deposits which suggest a sequence of structures. Here is another one where the deposits suggest, or the non-deposits suggest, a sequence of structures. What is nice about this, at the eye, you can see the cornea, you can see these three layers of the eye, which represents a structure like this, characteristic of modern-day crustaceans or malacostricans. Looking more closely, then you can see that there are 1, 2, 3 iterated structures in the head that extend then towards what should be here a brain, but is not visible. So this suggests 3 nested optic neuropils. But it's not quite good enough until one actually comes across the crown jewels, which before I show you the crown jewels, I'll show you these, which are eye stalks. We have a whole collection of eye stalks in various postures and various rotations, suggesting this animal indulged in active vision, like a modern shrimp or a modern lobster. So we have a huge number of these various attitudes, and the eye stalk posture suggests active vision, searching vision, and convergence. So this was no simple visual system. And then we found this. And this really was extraordinarily exciting. In fact, it was one of the collection in the Yunnan Key Laboratory for Paleontology that had been written up before by Jan Bergström in Sweden. And he identified this structure here and said, this might be brain. But in fact, it's much more than might be brain. You have the incoming axons of the antenna. You have an incoming, sorry, here it is from the antenna. You have an incoming axon from the optic stalk. And you have another axon here, down here, coming up from another appendage, which would be the second antenna. So the entrance points of these various axons suggest a fusion of the 3 ganglia of the brain, the pro, deuto, and tritocerebrum. What is absolutely fascinating are these structures here, which are the 3 representations of fossilized neuropil. So here then is the 1, 2, 3 of the neuropil, here is the brain, and in fact there's a rough area here, very interesting morphological distinction of 2 areas here which correspond in a modern crustacean to the antennal lobes, the olfactory lobes. So Fuchsiana shows you unambiguous evidence of an advanced malacostracan-like brain. It doesn't show anything like a branchiopod brain with 2 neuropils, it shows a brain with 3 neuropils. So the brains of Drosophila, which have 3 neuropils plus a lobular plate, are by no means recent innovations. This organization is nothing new. It is very, very ancient indeed. So you have the 3 neuropils, you have your optic tract, you have a projection of the optic tract then into the dorsal protocerebrum. You have the projection from the antennae into the what is to be the deutocerebrum, the second segment, and then you have this little stumpy antennal 2 nerve which plunges down ventrally in the fossil and you can't follow it any further. And if you compare this and this, then they're very similar actually. This is the brain of Cenobita clipiatus, the land hermit crab that we've been working on. And you have the 1, 2, 3 optic neuropils plus this thing here, which is the homologue of the mushroom body, which we discussed yesterday. And maybe one could, stretch one's imagination a little bit and say this bump here might be then equivalent to the hemiellipsoid body in Fuxian Hui. And this is a dissection of a glass shrimp, a little tiny glass shrimp, the width of which was the same as the optic capsule of Fuxian Hui, showing also 1, 2, 3 optic centers, 1, 2, 3 optic centers, and then the medial protocerebrum and deutocerebrum of the brain. So that's nice. It's going to come out in Nature in October, I hope. This is a fossilized brain, and the nice thing about this is it does show these 3 nested neuropils. So this is pretty good. I would say the best one can get. It's almost proof that the branchiopod type organization certainly cannot be the ancestral ground pattern of what we see in insects and modern crustaceans. Not only that, the first branchiopods actually appeared on the scene about 480 million years ago, not 535 million years ago. Yeah.
technical question. The deposits that you find in the fossils.
Yes.
What are they exactly?
They have a lot of iron, some silicon, of course, everywhere, but iron, mainly iron. And my colleagues in National History Museum in London have been looking at elemental backscatter and using electron microscopy and these nice techniques. And I don't have the images here; they'll appear in the paper, showing the actual texture of the fossilized tissue versus other parts of the head, and showing this very, very high iron peak. Mm-hmm. These stains of brains in fossils are really mysterious. You know, you can see this organization as the negative image in this animal, and in fact, I was looking at this in the days before we found the other fossil, and thinking that I could certainly see these three regions, which don't seem to actually have any kind of deposits in them. They look like an endocast, and yet then you have this popping up in the collection, and clearly it looks like a brain that's been stained with Indian ink. Right. We need to go back and look at more, because there's the other trajectory which gives rise to the chelicerates. We think we have a different kind of brain in those.
Because I was wondering whether, if you would look in the specifics of these deposits and their density, you might again be able to distinguish the cell bodies from different kinds. I mean,
that would be
the density of the material you would think correlate to then this deposit.
Yes.
I was wondering whether you found such a correlation.
Well, we don't see anything that looks like cellular material. It is pretty homogeneous.
The density of the deposit.
Yes. Well, we do see different densities, and you can't really. I think this is not quite in focus. This image. There are two different. Two areas here of quite distinctive densities. And these areas correspond to the antennal lobe. So we would like to see maybe stratifications in these structures, but in this particular creature, we don't see it. There's too much to hope for. So it really is the crown jewels right now. It is a unique specimen, but it's an amazing specimen, and it's really amazing that you can actually then travel back in time, literally back in time, and have a really reasonable, well, have a good understanding of the basic organization of the neuropils in an animal which is incredibly simple. It's not a complicated animal with lots of different limbs and so forth, like Drosophila or a crab. It's amazingly simple, this creature, this Fuxianhui. So it's a simple organism with very, very nice eyes, active vision, and quite a substantial brain. Yeah.
Why should invertebrate brains fossilize and not vertebrate ones?
Oh, who knows? That's a good question. I think one of the reasons is that the conditions of the taphonomic conditions here are really exceptional. It was anoxic, it was very rapid. These are soft-bodied animals, so they were perfused very, very quickly with mineral salts in anoxic conditions. So the bacterial decay was minimal. There are fish at that level in 535 million that Simon Comey Morris has described, and I suspect must be brains in those. Yeah.
Because they have been found in this fauna.
That's right.
Fish-like animals, but not
fish-like animals.
Heads. So
there are cyclostome-like animals that have been described.
The ones that I've heard of are craniates, but the fossils are annoyingly missing bits of the head.
There are two that have been described with head attached with gills,
right,
and with nice eyes too. Very, very nice single lens eyes. I mean, they're great.
Right.
And one needs to look at those again, revisit them, and with the appropriate search image.
Yes.
I mean, it's like Srini was discussing yesterday, training bees to develop search images and finding shapes within patterns of random dots, very, very covert shapes. So you have to train them onto simple shapes in order for them to do that. You really need to have the search image here, ready in one's mind, to be able to pick this out. So Jan Bergström, when he described this fossil, he didn't have the search image. He just said there's a blob there. It was possibly brain. But if you have the search image, then you can actually see these structures, or you can pick them up very, very quickly. So yes, the problem is when you're working with the Chinese groups, these various laboratories are highly competitive, And Fuxianhui is in Kunming, but the vertebrates are up in Xi'an. And Xi'an and Kunming really don't speak to each other at all, so to get back up to the vertebrate fossils is going to actually require a lot of diplomatic, acrobatics. I don't know how to do it. Yes. But yeah, I'm sure there are fossilized brains of some of these earliest acraniate, agnathan, fish-like things. They don't look like Amphiopterus. They look much more like cyclostomes, like lampreys. That'd be great, wouldn't it?
But now, Nick, so the conclusion, I mean, your information is, so this is now at the origin of a whole set of taxa.
No, we're way off the origin still.
Okay, but this is my question. So,
Yep.
so how far are we?
Well, I think we have to go down at least another 30 million years, down to about 600 million maybe, at the interface of the Vendian and Cambrian. The trouble is that there's a small shelly fossils have been described. The first arthropod-like animals were the trilobites, the fossilized animals, and no trilobite has revealed anything about the nervous system.
But at this stage in evolution, how many basic, let's say, bowplants do you think exist now in parallel?
Oh,
This is one
none.
of how many?
I think there are two bowplans only.
Okay.
One that gives rise to the insects, malacostrogons, centipedes, and the other that gives rise to the chelicerates, the spiders.
Okay.
It's like having two. How did I say? The bones of the system of is the nervous system. Everything else is decoration.
Okay.
So you attack, you evolve a specialized limb, and that ganglion then remains. The bowplan remains, but it's modified to serve that particular set of sensory attributes.
So we're sitting here already just after the bifurcation.
Well, we're somewhat after the back. We have to go down deeper.
Yeah.
And we're now looking at the lower pods. My colleague in London, Xiaoyan Ma, she's got a lot of lower pod material. And lower pods show segmental ganglia, but the brain is really, really tough to get at. There's an apical mouth in the lower pods, and you can see something across the top of the mouth, but it's very hard to be sure of what it is. And there's no modern taxa which would provide a proxy of that organization. So this is for the future, and so I'm set to go back to China next year and do a lot more searching. Eventually we'll find them, I think, if we have the search image, right?
Right, exactly.
Yes. Yeah.
Wow, quite an adventure.
It is, and then of course one wants to find even deeper evidence, which would then unify both the deuterostome protostome trajectories.
Right.
But that's probably too much to hope for. I think that's it. As I said, this is just entertainment today. So really the purpose of comparative anatomy is to resolve the ground pattern. So try to resolve the ground pattern across different species. And then you can use phylogenetics, neural and molecular markers to reconstruct the tree and then interrogate it, and then at least make predictions about the likely ancestral brain organization. And then the convincing proof requires that we get into one of H.G. Wells's machines and travel back in time, but we can't do that. And if we did, we'd be very dangerous because we have the butterfly effect. So we have to go to these incredibly rare fossils and see whether we can actually find something that whets the appetite. And occasionally, we find something really quite extraordinary. And it's purely serendipitous, And I think these results would actually please H.G. Wells and Charles Darwin. And I must say, they certainly thrilled us. Okay, thank you very much for your attention, and that's it. I've talked too much.
An optimotor response that was in the direction of motion of the drum for a while and then
Yes.
reversed. What do you think is going on there? Is it suddenly switching attention from neurons in one preferred direction to neurons in the other preferred direction?
I think what it's doing is doing what it's always doing, which is searching for something to eat. And it's switching. Yes, it's switching, but it's following the stripes, and then it's not moving in any other direction because it's in an arena.
Right.
It could move across. It could search other parts, but it's actually either following the drum or moving the opposite direction.
It's not turning on a dime. It's not rotating in one spot.
It's actually turning around and going back again. Yeah. I hoped that it would, you would just switch and they would turn around and follow the drum, and when we reverse the drum. But I think it's looking for motion. David O'Carroll wants sometime to poke into the brains of these and see if he can record motion-sensitive wide-field neurons. And the wide-field neurons are there, as you've seen. It'd be neat to go, but I think one has to go to the tropics and get some of these very big creatures. Or on the island of Madeira, apparently there are quite large scutigeromorphs. So it's another reason to go to Madeira. But of course they've probably been burnt in the fires. But there are these very large animals, and they probably have, rather larger brains, one would hope, than the ones I got in Italy.
In terms of attention, you mentioned attention, why don't you just think about habituation? So for when you put them in the drum,
yes,
would all of them always initially just follow, let's say, with the movement of the rotation or against it, and then switching it?
That's a very good point. We've actually only tested 3 animals. We can only put them in singly, otherwise they start eating each other. They more often than not follow the stripes at first and then reversed.
So
I'm thinking the same thing.
Yeah.
Attention, selective attention is what I'm thinking of,
Yeah.
not habituation.
that's something not saying habituation to do the job.
Could do the job, do you think?
Yeah, sure.
Well,
But it's very fast habituation. It's in less than a minute. And they're going this way, and then they're going that way, and then they're turning back again.
to switch the habituated neuron that had to produce a lower response than the other neuron.
Yeah.
And that's kind of hard, I think. Really, it's the switching of selective attention. It's
Yes.
that's
Yes. They're impressive animals, actually.
Right.
They're terrifying looking things. If you have, to make a video of these close up. There's a fellow up in Greifswald working with Stefan Harsch called Andy Somke, who's sort of in love with these monstrous creatures, and he's got videos of these animals rearing up. But you look at the scanning electromyograph, that head, I mean, it's pretty intimidating, really. Now, we don't know really where these centipedes first appeared in the fossil record. There's very, very, very poor fossilization. So these small insects and centipedes are almost impossible to find as fossils in terrestrial deposits. They're known to be around in the early Devonian and late Silurian, but they must have appeared much earlier than that. And they think they represent even a more ancient organization than these 3 nested neuropils. But again, we have to search. This pretty animal, where is it? This one that is so very attractive. Why isn't it going back again? It doesn't like me. So this guy here with a very long reiterated body. It's very centipede-like. I would love to find the brain of one of these. It might tell one something important.
But it has quite some specialization, right? It doesn't have the sort of...
This guy.
Yeah, okay.
It has some specialization, but all the other segments are homonymous, like that of a centipede. It's just the first maybe 2 segments have got specialized appendage-like structures, one of them being the eye stalks.
Right.
It is even simpler than Fuxianhui. But we don't have the brain. We're doing experiments to try to embed this mudstone in plastics so we can start polishing them down. Yeah.
Is that promising? Does that look like a promising approach?
We have no idea yet.
Okay.
We haven't got the right plastic yet. So the plastics we use so far haven't been very successful. We're just using on sample stone. But there probably is some very hard acrylic stuff we could use.
Right.
Yeah.
So in these Chinese fossil fauna, so you get down to the lower Cambrian layer.
Yes.
And then what happens when you keep going? Is there anything?
Then there's nothing, and then you have all this strange Ediacaran stuff.
So there's a big gap. And
Yes,
but
yes.
If this was such what is the current theory? Because I read 10 years ago that people were talking about oxygen concentrations, things like this.
Yeah.
I mean, is there
That's one of the theories.
Yeah.
Yes.
But
snowball Earth and such like.
Yeah.
But I think all these theories, they're founded on studies of mineralization.
Yeah.
in the strata. But I don't know. I mean, it's really very strange. There seems to be nothing that's sort of gone across this barrier. There are various fossils in the Ediacaran fauna, things like Sprigina, that people have claimed are arthropods, but they're all asymmetric. They're not bilaterally symmetric. They look much more like flattened sea pans.
But there are trace fossils.
There are trace fossils, and I was talking about those yesterday. And they're very, very interesting, because the trace fossils do suggest worm-like creatures, bilateralians, which are using search strategies to find food and making choices, and also returning maybe to a previous spot, which suggests they have place memory. So those are very fascinating, but there's absolutely no fossil yet found that would correspond to the tracks it's made, which is a shame. But, you know, there are new things being discovered in Spain, up in northwest of Salamanca. There are lower mid-Cambrian deposits have been discovered, and there's a group. Actually, Garcia Villedo's son is Villedo Jr. He's looking at these deposits now and getting some really nice material out of them. So there are new deposits being found all the time, and around Kunming, within a 100-mile radius, there are 40 sites that one can look at.
So it could be some change in the sort of body parts of these things that have got wider bodies.
Yeah. One just has to look.
Time-wise, how big is this gap, roughly?
Well, you mean between the first appearance of arthropod-like creatures and the end of the Vendian? It's about 30 million, maybe more. It's quite a. Well, it's not very long in geological time.
It's still substantial.
Yeah. But the interesting thing for me is that there might be just these two ground patterns of nervous systems that today we see in the chelicerates on the one hand, and then the insects and crustaceans on the other, and that this has diverged very, very early in evolutionary history. So all these, the whole Cambrian explosion can be interpreted as not an explosion at all. It's two light motifs, and then there are all these sort of baubles hung on them as you go through evolutionary time. I've always been wary of this hypothesis of the explosion. It doesn't seem to make any sense. Although my wife suggests that maybe people from planet Zog emptied their bilge tanks 600 million years ago, and we're the result. Yeah.
You probably said this already, but can you reiterate the difference between these two plants, the spiders and the...
Can we what?
Can you reiterate the difference between the two plants?
Yes. So you'll be pleased to hear that the chelicerate visual system, which has also got color and achromatic pathways, these are totally separated in two different neuropils, serving two different lens systems. And the organization of the neuropils, although there are 3 neuropils, is utterly different from any crustacean or any insect. And so when you interrogate the cladograms, it shows you what you suspect, that this is a very nice example of convergent evolution. So the earliest nervous systems of the chelicerates probably did not have these three successive optic neuropils. They must have had a very, very different shaped brain. And also in the chelicerates, the fusion of the first, second, and third ganglia is much more profound than in anything except very, very recent insects and crustaceans. And that probably is also an early event if you look at limulus, for example. So there are morphological distinctions, I think, which provide a good search image to look for these brains in trilobites and in things like Naroa. I showed you Naroa as one of the, let's see if I can find it, one moment. Naroa is this creature here. So this is the one we're really, really keen to look at in much more detail, this group. The Naroids, with big head shield, and they're very, very, very trilobite looking. They remind one of limulus too. So those are the ones that are going to be the next target for our studies, to see if we can find those brains. Yeah.
Other questions? Well,
That's it.
Thank Nick for this feast of lectures. It's pretty amazing.